Today, we will join Dr. Sonny Lee, Assistant Professor in the Division of Biology, talking about the identification of microbiome communities and core function of microbiota in healthy pigs across the stages of production. Dr. Lee?
Thank you, Maureen. So my project for the global food systems was identification of the microbiome in terms of the microbiome communities and their function, especially concentrating on the core function in healthy pigs across different stages of production. Before I go on to talk a bit more about the introductions and some of the results we have achieved so far in the last 2 years and still continue to explore the data, I'd like to thank some of the people who is involved here. And of course, my lab and some of the really good collaborators, whether in K-State or outside K-State, especially like to thank the Roof Lab, the Roof Farm, which has helped me to make this work possible. We have been working with them very closely throughout this whole 2 years. And in fact, we're also working, we have just met 3 weeks ago. We are thinking about what we are going to do next now that we have all this data, we have all these results, and I can tell you a bit more about what was our meeting about at the end of these 3 weeks. So this work started looking at the microbiota function throughout the lifetime of the swine. However, it has since evolved into quite a numerous work from using the swine as a model study for human disease to fecal microbial transplantation in a viral challenge and to antimicrobial resistant genes in working with colleagues and collaborators at VetMed. So this project itself has constantly been evolving and looking at more and more data and with more collaborators as we go along. So today I'm just going to talk about the project that was started working from the fundings, benefit from the fundings from GFS, and it was looking at the microbiomes in the lifetime of the swine. So there are 3 distinct stages in the pig: the pre-weaning stage, the nursery stage, and the growth stage. And within these 3 stages, the pigs are always under stress, whether is it diet and housing or whether is it their age. And constantly changing of the constant stress on these pigs changes the pigs in terms of the physiology, the health, and also in terms it changes the microbiome, and the microbiome again then in the vicious cycle or in a cycle affects the pig again, and this just keep going round and round. So in our research, we want to find out like how does the microbiome evolve through the stages, and how does this microbiome in terms of the membership and in terms of the functions changes as through the whole 3 stages. In terms of really broad perspective, we want to look at the microbes in terms of the bacteria, in terms of the fungi community, and going further, we want to look at the microbial potential functions. What are some of the essential microbial functions that enable this exchange between the microbiomes and their host? And hopefully moving further on that, we also want to look at the products of these microbes in terms of the microbial metabolites, speculating that these microbial metabolites are actually really, really important to the host health. How did we do this? So we started with 10 pigs, and we followed these 10 pigs throughout the 3 stages of the growth, from pre-weaning to nursery to the adult growth. Altogether, among the 3 stages, we collected 23 time points with a total of 234 samples. And we extracted the microbial DNA from the fecal samples, and from there we look at the microbiome in terms of the taxa using 16S and ITS marker genes, and using shotgun metagenomics to look at the function. What I depict here is only one farm, which is the K-State farm. On the other farm, which is a Roof farm, we did exactly the same. So altogether, we have a total of 468 samples. So that was quite a big undertaking for the lab students, and I am really grateful to them. And so far, we have done everything, and we are analyzing the data. What I'm going to talk about today is a very small portion of the data. Right? What you are looking at here is the bacterial community according to the age in both the K-state farm and the roof farm. So what you're looking at here, the orange, the yellow, and the red represents the microbial composition of the pigs coming out from the K-state farm, whereas the purple, the green, and the blue represents microbial communities coming out from the roof farm. Orange and purple represents pre-weaning. Yellow and green represents nursery. Red and blue represents adult pigs. So as you can see, it is interesting to us to observe that the pre-weaning of both the farms are actually very different. And what's more interesting is that as they grow further older, the adult pigs have very similar in terms of the microbiome composition. And well, one of the things that is interesting, but we kind of expected it, was that the microbiomes of all the pigs in terms of pre-weaning, nursery, of adults was actually a lot more similar to each other as compared to that in a commercial farm, right, a roof farm. And I think that is because of environmental factors, and in terms of a K-state farm, in a research farm, is a lot more well organized and a lot more well controlled as compared to the commercial farm. However, as you can see, even in a commercial farm, as the pig becomes older, the microbiomes among each other, among the 10 pigs, starting to get even closer to each other. When we are looking at the microbiome of the pig, of course, we want to know about the diversity. Here, what you're looking at is bacterial diversity in all the pigs, whether is it in roof farm or the K-state farm. The red represents pre-weaning, blue represents nursery, and green represents growth. And the index, Shannon index, as it increases, means it becomes a bit more stable. As it's low, it becomes, the diversity is lower. So as you can see in the pre-weaning, the diversity of the pre-weaning pigs were very low, and it started to increase very fast to a stage where it started to stabilize during the nursery stage. And there's a bit of increase during the nursery stage overall, but at the growth stage, there's not much increase anymore, and the diversity of the microbiome is really stable. While we're looking at the diversity, we also want to understand what about the volatility. Volatility is how much changes in those pigs among the age and how much differences between one individual to another. You can see there's a line here which marks 0. And when the graph reaches 0, it means that there's no volatility. It means that there's really stable amongst the microbiome. Again, red represents pre-weaning, blue represents nursery, and green represents adult growth. As you can see, in the pre-weaning pigs, the microbiome is really far from the 0 point, which means that it is very volatile. Things are always changing from one day to another. Things are different from one pig to another. And as it approaches the nursery and growth stage, it gets more and more stable as it goes past through, as the pig gets older. So putting all together, all this data kind of shows, collaborates with what we've seen in the clustering plots, as in like the pre-weaning stage. The pigs microbiomes are very different, and they started to converge at the adult growth stage. So of course, we want to know who are there in these pigs. And what we did was we identified 3 distinct phylums, and they are we call them the core phylum, the transition phylum, and the increasing phylum. What you are looking at here is each of these rows represents one phylum, and each of these columns represents one sample. And they are kind of clustered according to the pre-weaning, nursery, and adult stages. As you can see, in terms of the core, most of the phylum, most of the bacterial populations exist throughout the whole stages of the pigs, whereas in the increasing, they were more lacking in the pre-weaning, and they started to show up a lot more, increasing in relative abundance at the growth stage of the pig, and so on and so forth. So we are still going through all these data and deciphering what is meant in terms of some of the taxa that is going up and some of the taxa that is decreasing as the the pig starts to grow through. However, we were thinking that what we need to do is we need to evaluate the genes or the microbiome functions to understand that in terms of the core transitions or the increasing clusters, what are these bacterial population actually doing in the guts of these pigs, and how does it affect the pigs, and how does the pig affect the microbiome, and vice versa. So as you can see, a quick bacterial review is that one, there's a distinct difference in the pre-weaning nursery pigs in both the farms, whereas in the adult stage, the both farms seems to converge together. The bacterial populations, in terms of diversity, starts to increase during the developmental stage, and it gets really stable at the end part of the nursery and the adult stage. There is a distinct pattern in terms of the phylums that follows throughout the lifetimes of the swine. And of course, when we come to this point, we realize that although this diversity and taxonomic information are really useful to us, we still like to answer what are all these microbes doing? What are they performing? What functions are they performing, and how they affect the swine host health and well-being? As you can see at this stage, the project kind of getting into two directions. One is just looking at the taxa, who is there, and the other one is where we are starting to look at what are they doing in the guts of the host. So we have, and I'm really lucky to have really good undergraduate students in the lab who is taken the role of analyzing the taxonomic and diversity information. And one of the students even got a Tracy Chapman Award just to investigate this, and I think she's making some headways. Whereas on the other hand, the microbiome function is undertaken by a graduate student, and she is also taken quite a lead on that as well at this present moment. So the next, I'm going to talk just a bit about the microbial function. Altogether, we selected a subset of the samples to look at the functions. And we did shotgun metagenomics and come up with a whopping of nearly 4 terabytes of data, and took almost a year to assemble these genomes to find out who are these genomes, who are these bacterial population in there. In terms of looking at the microbial function, what I'm presenting to you here are mostly from K-State Farm, because in terms of the roof farm, we are still in the very early stage of analyzing. So what are we looking at here? What are they potentially looking? Here, each of these rows represents one sample, and each of these columns, or I call them the splits, represents what we call a metagenome assembled genomes, which they assemble genomes from a whole set of shotgun metagenomic information. And in layman terms, we will call them bacterial populations. So as they are darker means that they exist more, and if they are lighter means that they are detected less in those samples. As you can see here, they have a very distinct detection whether they are in the early stage of the pig or whether in the later stage of the pig. So what does this give us besides just how they are detected? It also gives us the functions within itself. Again, looking at this now, instead of each of these splits, instead of a metagenome assembled genome or bacterial population, it represents one single function, right? So what you have here is actually one bacterial population, which is the single small split that you've taken out from the previous figure. And each of these rows again now represents one sample within all the 7 pigs or 100 over samples that we've taken as a subset for the metagenome sequencing. The green represents the pre-weaning samples, the blue represents nursery samples, and the red represents the growth adult samples. Again, each of these splits or columns represents a single gene function. And looking at it, you can see that most of these gene functions in this bacterial population is detected in the pre-weaning. It's barely detected in the nursery pigs, and is almost never detected in the adult pigs. And what are these gene functions? And from here, I can actually detect that if you look at the very outside of the circle, there are some black lines. Each of these black lines represents a carbon utilization potential, which means that this pig or this bacterial population that is highly detected in pre-weaning also has a lot of carbon utilization gene function in this bacterial population. And so this goes on to make a long, tedious process of how does this relate to the pig in the early growing stage of the pig. For example, another type is down here, you're looking at only 4 layers in the pre-weaning, because the other 660 layers, they have no detection at all in terms of this MAC28 or this bacterial population 28. Here, I just flipped the circle into a horizontal point, and each of these columns again represents one gene function, and each of this row represents a sample. And the red color here points out to the gene functions that is related to bile acid tolerance. And here we were speculating, how does this bacterial population that exists in only the pre-weaning samples are able to tolerate a high bile acids environment in the pig itself? Looking at it on the very big picture, and what we have here is we took every single bacterial population that we have assembled, and this is only a small subset of it, and we blast it to every single database that we have available that we can download, from carbohydrate metabolism, nitrogen metabolism, sulfur metabolism, to all the way to short chain fatty acids and alcohol conversions. And just for shit and giggles, we also blast it to photosynthesis database. And this photosynthesis database for us is actually a control, because we're hoping that nothing shows up. Because if anything shows up here, maybe we need to go back and redo some of the analysis. So moving forward, what do we do next is we have collaborators and various partners from K-States in VetMed to chemical engineering, where we are trying to come up with ways of looking at micro interactions and microbe-host interactions. And we also have collaborators with USDA Agriculture Research Division and University of Chicago to look at the metabolisms and the metabolites on the pathways. So my last message for today was that this is a really exciting time to do research, and we have a lot more data in a single study than we can handle. And this is the time that we put together, or at least what we in our lab, we are putting together new tools and traditional skills to answer those questions that has been like eluding us for the longest time. And with Ruth Lett, as I mentioned in the very beginning, we had a meeting 3 weeks ago. We were talking about the swine farm holobiome. They were interested in this because they were looking into the possibility of producing crops on their farm, and they wanted to know how does the swine microbiome, whether you will or will not affect these crops and the irrigation system if you are setting that up. And with that, thank you very much for GFS. Thank you for the support in these few years and continued support. Thank you.